This evening, I'd like to introduce you all to the Ediacaran period, which is the most recently defined period in the geological record, and also contains a large variety of very weird and wonderful fossils and organisms. So what I intend to do is give you a general overview of the Ediacaran period and the organisms found within it, and also try and convince you that their evolution is one of the key stages in the development of modern marine ecosystems. So if we go to modern marine ecosystems today, what we see is a diverse range of environments and ecological niches. For example, in the water column itself, we have a variety of organisms, macroscopic, large scale, such as sharks and fish, also microscopic scale, plankton. Plenty of diversity within those groups alone. And on the benthic environments, such as the deep sea floor, we also see a large amount of diversity in terms of organisms and a variety of different phyla and kingdoms. These two bottom pictures, they're both from Antarctic ecosystems, and you can see they're very different in terms of the organisms they're preserving. Those are different ecological niches in a very similar environment. And what I'm trying to suggest to you is that these very diverse modern ecosystems that we see today haven't always been this way. Again, another example of the range of organisms we see in modern marine settings. Not just inclusive of animals, we also see things like kelp forests, for example, but amongst the animals themselves, a wide range of body plans and ecological adaptations to survive in certain particular environments, both amongst vertebrates and invertebrates. So if we look back into the fossil record, what do we actually see? This piece of limestone is of Wenlock age, so 430 million years old in the Silurian period, and it comes from Dudley, which is not too far from where I originate myself. And it contains a wide variety of fossilized organisms that are all very similar to the things that we see alive today. So there, for example, is a brachiopod. We have sponges. We have little bits of arthropod. Arthropods, when they die, often fall apart, and so we don't see complete preserved specimens very often. And evidence for things like crinoid stems and ossicles. And if we look for elsewhere in the geological record, we also see evidence of organisms seen today, both on the small scale, such as foraminifera, all the way up to the largest organisms known in the oceans, whales. So going back through geological time, looking first at the Phanerozoic. Phanerozoic means visible life and is a hangover from the period in the 1800s when people were naming these geological time periods and couldn't find any fossils anywhere below what is now the Cambrian period. So looking back in the Mesozoic era, around the Jurassic, when all the dinosaurs were around, we see a variety of organisms that we can relate to. We see organisms with a dolphin-like body plan, here ichthyosaurs, marine reptiles, but taking advantage of the same sort of ecosystems and lifestyles that modern dolphins engage in. Rather than having squid and cuttlefish, we see very similar relatives, also cephalopods. These are the ammonites. And we also see evidence, instead of coral reefs, we see reefs that are made up of organisms that are bivalves and not at all corals. Even further back, twice as old now, around 400 million years ago in the Phanerozoic, again we can still see fossilized evidence of organisms we're quite familiar with, from the brachiopods I showed earlier and the crinoids to fish and trilobites, which are arthropods. And going as far back as we can into the Cambrian, in terms of hard parts, so shells and bony teeth and skeletons, we see bits and pieces of organisms that we can relate to. This, up at the top of the screen, is a mollusk shell. Not at all similar to any modern mollusks, but you can still recognize it as being something related to a snail. And spicules that suggest the presence of sponges. But we also begin to see a variety of organisms that are very different and very strange. This one is called Halkieria. It comes from Greenland. And the two shells that either end of the organism are thought to suggest that this might be a primitive bivalve. But we're lucky in the Cambrian that we also get very good preservation of soft-bodied organisms. Here's an example of a worm from Chenjiang in the Middle Cambrian, and these two examples are from the Burgess Shale in Canada. And although this is an arthropod, and we can tell that from the carapace, it has a segmented body plan, it also has a lot of strange body features such as 5 eyes and this weird appendage, trunk-like appendage on the front. It's very different to anything we see in modern environments. And we're also lucky enough to be able to study microfossils. So here are some teeth of various annelid worms and mollusks that have been extracted from rocks by colleagues I have at Cambridge. And if we were to try and work out what these ecosystems look like in the Cambrian, based on the hard parts, or the organisms with hard parts alone, we might consider them as quite dull, with very little diversity, little evidence for tiering. That's utilisation of things other than the very bottom levels of the seafloor. But if we consider all of the soft-bodied organisms as well, this view changes, and what we actually see is an environment where the ecosystems are very similar in diversity to the most diverse ecosystems seen today. However, for the bulk of the period that life has been present on Earth, almost 3 billion years, ecosystems would have looked very different, almost barren, because there's no macroscopic organisms. At most, we will have seen things called stromatolites, which are layers of microbial growth, bacterial growth, that form today in environments such as Shark Bay in Western Australia, where you have no predators able to live in the saline conditions that could eat through those microbial mats. And microbial mats is something we'll come back to later. It's a very important part of the story. Before the Cambrian, rather than the macrofossils, we see microscopic organisms. And here is a column we've been looking at so far, the Phanerozoic. That fits into this very small green section in the history of the Earth itself, right at the top. Back down here, at 3.6 billion years ago, we see the first evidence for life in the form of small spheres and rod-shaped bacteria. This scale bar here is 10 micrometers, so 1/100th of a millimeter in scale, so very small. As we move through the main evolutionary developments, around 1.8 billion years ago, we get a large increase in size of the microfossils we're seeing, and some new diversity. Fungi appear for the first time, and acritarchs, which are thought to be encystement structures, so egg cases of various different planktonic groups. And then the first eukaryotes emerged, multicellular organisms, around 1 billion years ago. These examples are from the Torridon in Scotland. The story really begins to pick up in terms of looking for modern organisms in the Cryogenian period, which is around 750 million years ago, when we see in the plankton the first evidence of organisms that could form hard parts, so biomineralizing. These include various ciliates, foraminifera, that many of you might be familiar with, and amoebae, so all very small, simple organisms, but starting to develop external structures. And most of these are just, it's called agglutinating, where they stick grains of sediments to themselves from the water column. In terms of large fossils, we actually see the first that are visible with the naked eye. These small, simple discoidal things that you see at the top are probable microbial colonies growing on the surfaces, and they come from Kazakhstan and Russia. So we go from this large period of Earth history where all we have is microbial organisms, and into this Phanerozoic where we see all the modern ecosystems and modern organisms we can relate to, and in between that is the Ediacaran period, which is the focus of this talk. And there we find a transitionary stage where we see the first large and complex macroscopic organisms but they're not things that we can easily put into categories of organisms that are alive today. Now, the Ediacaran to Cambrian transition is probably the most important transition in the history of life in terms of the number of innovations that were present and evolved at this time. So, for example, it's the first time where we see macroscopic biomineralization. The plankton had started doing this maybe 150 million years before, but actually in large organisms, the development of shells, teeth, hard parts. We see the first evidence for predation and for guts, through guts, which allow for processing of sediment and also for organisms to prey on one another and start up arms races, evolutionary arms races. We have the development of modern ecosystems, but the three things I think are most critical, and that's what I'll focus on in this talk, are the first evidence for animals themselves, the first evidence for locomotion on the surfaces and therefore muscular tissue within these assemblages, and then continuing on from that, the first evidence for biotubation, that is vertical burrowing and therefore mixing of the sediment at this time. And those three features I'll cover in a lot more depth and also try and show you how their evolution impacted on oxygenation of the sediments and therefore how habitable these ecosystems were, and also the decline of microbial mats and how that relates to the fossil record itself. So the Ediacaran period in brief is found here around 90 million years long at the base of the Phanerozoic, just below the Cambrian. Its base is defined by a limestone unit in South Australia, whereas its top is defined by the first appearance of this small vertical trace fossil called Trepticnespedum, and that's found in Newfoundland in Canada. Actually, what you don't see in this picture is just over to the side is the town's rubbish dump. It's not a very sensible place to have put a major geological boundary, but that's the way it's gone. Now, this point in time is also a time where we get, as well as the biological transitions, we get very large-scale physical and climatic events. The first of these is a series of global glaciations, and they're marked by the presence on many continents at similar times of three sets of mixed rocks. They're called diamictites, which means mixed rock. And as you can see, my glamorous field assistant Joe Stewart here is pointing out large clasts within these, and if you look closely at many of these clasts, you can see a deformation in the layering of the rocks units beneath these clasts, which suggests that these large lumps of rock were dropped in from above and therefore are likely to be ice rafted. So as you have a glacier moving off the edge of a continent, as it melts, all of the material it's eroded off is dropped into the sediment beneath. Now, these deposits are found globally. They can be kilometers thick, and they led to the idea that at certain points in Earth history, the whole planet was frozen over, and this is called the snowball Earth hypothesis. Now, in recent years, the main proponents of that hypothesis have started to accept the arguments of other scientists in the field who suggest that maybe a snowball Earth is far too severe an event for life to have actually survived several times in Earth history. And so this new model, where only part of the Earth is iced over, called the slush ball model, is now probably what is most accepted. But needless to say, there are these three large glacial events at this point in time, and they will have had a significant impact on life and the biosphere. We also have, ignore the data points here, it's just to show you large excursions and changes in the chemistry of the oceans. These are carbon isotope records, and these are some of the largest excursions in history, which suggests that the carbon cycle at this point is very different to that seen in modern settings, and very different to what was seen before the glaciations. We also see a major change in oxygenation and oxygen levels on the planet. So this is one proxy for, or one way of measuring oxygen levels. You can't unfortunately measure them directly from the rock record, but looking at iron ratios within sediments of this age, And what these authors are suggesting is that there's a switch from values which would suggest there's no oxygen in these ecosystems for the end of the final glaciation, which is this Gaskier's unit here, and a switch to oxic levels just afterwards. And this, the Druick formation here, is where you see the first large Ediacaran macrofossils. And so there is a coincident link between these two events. Whether that is a real link and the oxygen is responsible for the evolution of these organisms is one of the main questions in Ediacaran research. And finally, there's some very large changes in tectonics. Just up to the start of the Ediacaran period, we had the presence of a supercontinent called Rodinia, which consisted of most of the major land masses on Earth. And from around 700 million years ago, it started to break up and rift. And you can see some of these rifted blocks of continents from margin moving away from the main area. And what that does is provide you a lot more surface area around the continents of shallow water settings, where nutrients, the weathering off these newly formed continents, can enter the marine realm in the oceans and produce blooms of algal organisms and also drive evolutionary diversity. So, the Ediacaran period, as I've mentioned, is considered to be the time when the first animals appeared. And there's a lot of popular science books that have been written on this subject that hint at animals being what these strange and weird and wonderful organisms may represent. But it's not completely accepted that that is the case. And in more recent years, people have suggested that it's much more enigmatic what these organisms may be. Evidence for animals in the Ediacaran period largely follows from what we see in the fossil record. If you ignore everything else on this diagram other than the blue and the yellow blocks, as we're going through time from the Cryogenian through to the Ediacaran, there are no animal phyla or classes present. And suddenly, at the very base of the Cambrian, we see a rapid increase in them through the Cambrian. By the end of the Cambrian, we see most of the major classes and phyla of animals we see today represented in the fossil record. This is a very rapid and sudden change in biology and evolution. And if we look at the molecular data, so looking at the DNA and the genomes of all the major animal groups we know today, and try and trace back from those when their last common ancestor is likely to have been present, those data would suggest that most of the divergences, the splits between these different groups, such as the mollusks and the annelids, for example, would have been present and would have taken place in the Ediacaran period. And that suggests to us that we should be seeing ancestors of all these major animal groups back here in the Ediacaran, if these molecular clocks are correct. At present, we don't see that presence of organisms. And so what we do see instead is, in this green block here, a whole biota and assemblage of organisms is very different to anything seen before or since. This is one example of how hard it is to work out what these things are. This is an impression of one organism called Thectardis. As you can see, there's not much more to it than the basic triangular shape. There's no morphological internal structure, there's no detail to it, and somehow people have suggested that this might be one of the earliest sponges. It's very hard to draw information out of these fossils. So what do we know about Ediacaran organisms? Well, the first large complex things we do see are from China and their algal fossils. We're very confident that that's what these are centimetre scale bars, so fairly small centimetric organisms with filamentous structure preserved as carbon impressions on the rock phases are representing. You also see around 580 million years ago, so halfway through the Ediacaran period, what have been interpreted as animal embryos. These are very tiny spheres that are found again in China in the Dushantou Formation, and they're preserved in phosphates. And it's been suggested that on the basis of the fact that you can see what appears to be cell division, so you get 1 cell, 2, 4, 8, 16, etc., that these may be embryos that are being preserved. Unfortunately, there's a lot of work been going on with this, because if correct, these would be the first animal fossils. It does seem that things aren't quite as simple as that. There are thousands and thousands of these structures, and the 1, 2, 4, 8 cell divisions that are described in the initial papers are not the only ones that are there. You get 6, 7, lots of other different numbers, and you even see these peanut-shaped things, which seem to have multiple cells within them, and this is not what any modern animal embryos do when they're splitting and dividing. Therefore, it's been suggested that they might be non-metazoan, so non-animal organisms, still similar to embryos in the way that they're in the life cycle stages that they're preserving, but not animals themselves, some sort of simple protystan organism. So we come to the Ediacaran Biota themselves. Now, these are organisms that are soft-bodied. They're preserved as impressions on sandstone and mudstone units around the world, and in a similar way to how you would see a imprint preserved in a sand beach. They can reach up to 2 meters in length, and they're found in over 109, I think now, different localities around the world. You can see there's a variety of different shapes and structures to these, but that very few of them can be related easily to modern animal groups. You might notice the similarity here with this impression. This is Charnia masoni, discovered first in Leicestershire, in Charnwood Forest in England, with plants and fern fronds, for example. But the thing is, these are found in deep marine settings, below the photic zone, and therefore they won't have been able to photosynthesize. Therefore, although they look like plants, it's a morphological convergence. They weren't plants themselves. So these are some of the environments that they're found in around the world. The best examples are the green little dots here, and are found in Newfoundland in Canada, eastern Canada, Charnwood Forest in Leicestershire, the White Sea of Russia, the Ukraine, the Ural Mountains, far northern Siberia, China, Namibia, and quite a few in Australia. And the Australian sites in the Ediacaran Hills are where the Ediacaran period has got its name. So there's a wide variety of morphological complexity within the Ediacaran Biota. You can see organisms with flat sheets-like structures with what have been described as quilted body plans. These are thought by some authors to be inflated, like a little mattress or the Michelin Man you've seen in adverts. Some structures with quite striking symmetry, such as this Earandromeda, have been suggested to be maybe relatives to ctenophores or jellyfish, whereas others appear to have been preserved within the sediments and living within the sediments, but don't seem to have a front or back end, and they don't seem to have any evidence for morphological organs or tissue structure at all. So there's a lot of different types of creature there, and they've been suggested to be almost every type of organism possible. So the fronds we see have been suggested to be seaweeds. Some of the discoidal fossils we see have been suggested to be similar to microbial colonies. So this is a microbial colony found in France today, and these are some of the fossils that we see in the Ediacaran. Some authors have suggested they're an entirely extinct kingdom that was around in the Ediacaran and then was wiped out by the evolution of animals later in the Cambrian. And the same author has also suggested that they may instead be protists, similar to modern xenophyophores. And finally, lichens and fungi have also been suggested by other people, with the fruiting bodies of various fungal organisms suggested to be similar to the fronds. Now, there is evidence to support almost all of these claims, but the problem is most people looking at these have grouped all of the organisms together. So what we're actually seeing is probably a mixture of all of these different kingdoms and phyla of organisms being present in the Ediacaran, but not within one single group. And this is just a quick tree of life I put together, and these are the various points at which Ediacaran biota have been claimed to be along this tree. And even if you look at one simple group, the frondose rangeomorph organisms, which are in orange here, you can see that they occupy most of the positions on that tree, including the animals up here in the region. So in terms of evidence for real, true animals in the Ediacaran, around 555 million years ago, we see several examples of things which could well be modern animal groups. So evidence for possible sponges with what appear to be spicules coming off, radiating from them. The ctenophores, as I mentioned earlier. Possible cnidarians, not corals or jellyfish, but this time sea pens, which are found today in modern deep marine settings. Organisms which appear quite simply as discs, but appear to have a five-fold symmetry, star-shaped symmetry there, which would suggest maybe that they're related to echinoderms. And then certain very well preserved organisms from Russia that have been suggested to be early mollusks, and some scientists have even suggested they can see guts preserved within some of these structures. It's not entirely clear yet whether that's the case. But these are all found in this region here. These are those organisms around 555 million years ago. And for the purposes of this talk, I'm going to assume that most of those, or some of those, are likely to be true animals. There's details that people can argue about, but it does seem that there are animals within that. What I'm far more interested in is a large group of organisms living in this period around 20 million years before those that are currently not able to be assigned to any modern group. And those are called the rangeomorphs, and they're frondose organisms that look like ferns. They're found mainly in two regions, which are grouped together as the Avalon, because originally, in Ediacaran times, they were actually located right next to each other. It's only the opening at the Atlantic that has separated them. So central England and the eastern coast of Newfoundland in Canada. So this is a sort of field area that we get to work in with these. These are fossil-bearing bedding plains, and they're exposed like this because of volcanic ashes that have covered these organisms and seafloors at various points in time. So that ash there is the one we're looking at, that small, thin white unit, which is actually about a foot thick. And those ashes smothered the ecosystems, covered the organisms in life position, and preserved them exactly where they fell. Which means that on an area about this size of that bedding plane, there's around 4,000 different fossil impressions. So very diverse, very abundant. And they occur within this very thick sequence of almost 2,000 meters of turbidite sedimentology. So we're on a deep marine slope environment or basin. The fossils found in these Avalon regions all date to around 580 to 565 million years ago. They are the first large and macroscopic possible animal organisms in the fossil record. And they are all constructed of very similar units, architectures. So you can see this simple fern-like, frond-like structure is sometimes found with a stem and a disc anchoring the organism to the sediment. Sometimes you find multiple fronds arranged similarly to a lettuce or a cabbage. Other times you see the fronds with different arrangements of the branches but no stems or discs. And some of them are completely different, much larger, up to 2 meters, as I said. They're all deep marine and preserved, as I said, beneath these volcanic ashes. Now the rangeomorphs are all grouped together because they're each composed of these tiny rangeomorph elements, they're called. So this large fern-shaped branching structure you see is constructed of further smaller fern-shaped structures that are self-similar on smaller levels and scales. And as you can see here, this is one frond. It's about 3 centimeters. No, sorry, that's wrong. It's about 15 centimeters in length, and what we see is in yellow a frond-like structure constructing this. In purple you can see that that itself is constructed of another identical frond-like unit, and in red and in green These are self-similar on at least 5 orders of fractal branching. And there's no evidence for any pores within them. There's no evidence for any polyps or anything like that living amongst these branches. They are simply external impressions of the organisms. And they're very beautiful. This is a surface in Mistaken Point where you can actually get a guided tour and walk on a 565 million year old seafloor. It's a very magical and tactile experience. But there are problems with trying to interpret this. This is one of the bedding planes we studied on a very foggy, dull and misty day in Newfoundland, and unfortunately there's a lot of these every year. If you're lucky, you get a sunny day, and this is exactly the same area seen in the sun, and you can see that these things, because they're preserved with such low topography, low surface height, what you need is low-angled sunlight to be able to bring them out and see everything to full effect. And if you zoom in on some of these tiny structures on the bedding planes, you can see that there are tiny little Rangeomorph fronds being preserved, less than a centimetre in length, but remarkably still there. So were Rangeomorphs animals? This is probably the biggest question in Ediacaran paleontology at the moment, because if they are the ancestors of almost all animals alive today. It's very difficult, however, to say anything useful about them, because even when we find them preserved three-dimensionally, or seemingly three-dimensionally, all we see is the external structures and features. We don't see any evidence of internal structure or canals within the organisms. We don't see any evidence that they might be filter feeding or have any extra additional organs, body parts. And so we're limited in what we can say about them from shape and structure alone. One thing that does seem to be agreed on in the literature, at least, is how they were feeding. And as there's no evidence of filter feeding appendages, it's thought that they were osmotrophs, and that is absorbing dissolved organic carbon compounds from the seafloor through their external structures. So to summarise what we've learnt or seen so far, the Ediacaran period sees the first large and complex organisms in the geological record, and they occur around 600 to 580 million years ago. And although initially the assemblages are dominated by algae, they're followed by organisms that could potentially be the earliest animals. And by 560 million years ago, we probably do have real bona fide animals in the fossil record. But the first members of the Ediacaran Biota can't be assigned yet to any particular group. So how are we actually going to get to the bottom of that? There's several ways we could approach the question, and the one that I've used most recently is looking at evidence for their behaviour, and in particular for movements and locomotion on these surfaces. If we look at the record of trace fossils, so that is evidence for movement in the fossil record in the Ediacaran, a review in 2006 suggested that of the 3 main assemblages, this is based on time intervals within the Ediacaran, in the youngest, around 550 to 540 million years ago, there were plenty of trace fossils, lots of evidence for organisms moving. Around 550 to 60, we see small unbranched horizontal structures that could be trace fossils and suggests the presence of animals and muscular tissue organisms moving around. But in the Avalon region, the one that we've been focusing on, no evidence of trace fossils. Those younger examples include structures such as this, where we see evidence for a fossilized organism, in this case a Dickinsonia, one of those sheet-like structures I showed you earlier. But then behind it, several impressions that may be interpreted to be grazing traces, where the organism has moved along the surface, stopped on the microbial mat, eaten it, and then moved on, and then stopped for a while, eaten again, and carried on in that way, as shown by this cartoon. But it's been shown that you don't necessarily need muscular tissue or movement, active movement by an organism to produce this. This is a piece of seaweed here, which has been put in a tank in one of our marine tanks in Newfoundland and just wafted by a current. And every now and again, it's on a microbial mat, every now and again it's moved and been placed down again, but the microbial mat that was underneath it has died because it hasn't had access to light, nutrients. And you get these impressions embedded in the mat, which if you were to dump a load of sediment on top, would then be preserved as similar imprints to this. So maybe these are not true trace fossils or evidence of muscular locomotion. Slightly more convincing, we see possible evidence of grazing, where we have fossils. This is a Cambrian from the White Sea in Russia, which seem to be associated with scratch marks on the surface, again interpreted to be feeding on a microbial mat with some sort of rasping appendage. And more recently, even this year even, there have been claims that in Siberia there's evidence for vertical burrowing and locomotion. Now this would be a very significant departure from what is traditionally accepted in the fossil record, because it involves a lot more behavioural complexity to be able to burrow down through sediment. And so there's a strong possibility that these have been misinterpreted and may in fact be some of these small shelly fossils that I showed you earlier, just forming lag deposits. Here are those shelly fossils in Spain, and they look very similar to those seen and interpreted differently in Russia. So the point of this slide is really to emphasize that the claim that you're having movements by organisms this far back in the fossil record is a very big and important one. It pushes animal evolution back quite a significant ways, 5 to 10 million years before we see any body fossil evidence for true animals. And so everything needs to be scrutinized in a lot of detail. So the Avalonian, as I said, is thought to be completely sessile with no locomotion. But that changed around 3 years ago with the discovery of some trace fossils in Newfoundland, a very similar area to mistaken points which I showed you earlier. These again occur in turbidite deposits. Here's the bedding plane itself, and you have a sedimentary sequence which is capped by a very thin, 1 millimeter thick volcanic ash. Now that covers these impressions, and as you can see from the arrows, there's little discs on the surface, and these are led to by small curved structures with hemispheric indentations within them preserved on the surfaces. Are they biological though? The first thing you have to rule out, remembering that these are 565 million years ago and therefore 10 million years older than even the previously proposed evidence for locomotion, is that these are not biological at all. And so you have to rule out things like fractures in the rock surfaces, cleavage fractures they're called, and evidence that they might be tool marks of objects that have just been dragged by a current along the surface of the sediment. Now one thing you can look at when doing that is if you generally see tool marks being dragged by a current, the current is usually going in one direction, which means that most of your tool marks will be in that same direction. What we see instead with these traces is that they go in multiple directions on the bedding plane, and they appear to cross-cut. So here's a series of 3 of them. I'll use these very tacky little dots to emphasize the directions they're inferred to be moving in, and that they are not easily explained by abiological, non-biological interpretations. So what could they be? Before we look at animals for evidence of locomotion, you have to also look at what other organisms are capable of moving in the modern environments. And one of those are protozoans. These are called gromid foraminiferans, about up to 3 centimeters in diameter, I think, grape-like structures that are found in Caribbean environments today, and they've been filmed moving and leaving traces behind them on the sea floor. However, their traces are very simple. There's no evidence of the hemispheric structures in between, just simple grooves along the surfaces, and there are fossilized tracks similar to this, even back to 1.8 billion years ago, that could be explained by simple protozoan locomotion without the need for any muscular tissue. The thing with the traces we found in Newfoundland is that they do have these structures within them, and in modern settings you need an organism that is able to apply a force in a backward direction against the direction that it's actually moving in to displace that sediment and move itself. And there's a variety of organisms that can do that. One is snails, and there's a study that came out a few years ago showing that modern snails produce tracks that are found in Cambrian settings that are very similar. And what we did was look at sea anemones. And for anyone who's seen the tanks downstairs, you'll see that sea anemones are typically like hard substrates, rocks, for example. And once they've found a good rock to sit on, they'll stay still and not move very much. However, if you take a sea anemone off a rock and put it into a soupy, muddy substrate, and we did this with a few of them, then what it wants to do is find a surface it can attach to. And so it will actively move, and it moves fairly quickly. So these photos are taken about 15 seconds apart. And what you can see is that the anemone moves by inflating its basal disc, its pedal disc. It inflates it up to a certain size, sticks it down to the sediment, and then tries to drag itself forward to where it stuck that disc. Now in this case, with the photograph, it hasn't worked because it hasn't been able to stick. And you can see from the mucus that's spewing out the back of it that it's trying very hard to adhere to this sediment. But if you give it long enough, it manages to do it. And this is one of the traces that, or two of the traces that these sea anemones manage to create. What you can see is that they're almost identical to the things we see in Newfoundland, with these hemispherical ridges of sediment, all leading to, eventually, if you pick the anemone up and move it, a terminal disc at the end of the trace. So here's the one from Newfoundland. And what we suggest is that to produce these ridges in a trace fossil, you do need muscular tissue, which would suggest that although none of the organisms, the rangeomorph organisms present in these environments, can be linked to these trace fossils, there were animals present in the environment at this point in time. This just summarises what I've said, but the important thing is that the presence of locomotion potentially has very wide-reaching implications for benthic seafloor ecosystems. Now that's because in the Precambrian, the Proterozoic 3 billion years before we get all of this complexity, most of the seafloors were covered by microbial mats. This is a modern microbial mat here. They're very leathery structures actually, and you can see from the colour distinctions that they're graded and banded based on the different metabolisms and ways of feeding used by each of the microbial populations. And they're seen in the fossil record as these wrinkly sort of fabrics on surfaces. This one here is typically called elephant skin structure, because if any of you have ever been close to an elephant, you'll see that it's very similar. And they're very much responsible for the preservation of the soft-bodied organisms that we're seeing in the Ediacaran period, because when you cover them up with either an ash or a sand event bed, the microbes that are there are responsible for chemically lithifying and hardening the sediment immediately in their vicinity. And if there's an organism lying on top of them, that sediment will be lithified around the impression of that organism, and therefore will take a mould of the organism itself. So they're very important for fossilization of these things. Now what we see in the Ediacaran is these microbial mats before the appearance of locomotion, covering these surfaces, nothing grazing on them, and the organisms embedded within them, these fundose organisms. When we then get the animal-like organisms that can move around on the mats, they start eating the mats, breaking them down, and by the Cambrian period, these mats don't exist at all. There's so many organisms feeding on them, burrowing through them, mixing the sediments up, that you don't get preservation of the mats, and therefore you don't get the preservation of soft-bodied organisms, except in exceptional environments where you remove all of these burrowing organisms and change the chemistry. And that marks a fundamental shift in the way that the biosphere as a whole was structured, because before that in the Ediacaran you see organisms not moving around, completely sessile, but as soon as you start being able to move, burrow into sediments, you begin to oxygenate it. By mixing it, you break up the microbial and chemical boundaries and gradients within the sediment, allowing oxygen into there, pushing down all of these zones where previously fossilization could take place, and therefore permits, firstly, much more of a habitat for various ecosystems that organisms can live in, and also push away the zone where you can preserve these fossils. So to summarize then, the view from the 1970s looking at these fossils was that they were all animals, with the discs being jellyfish, the fronds being sea pen-like cnidarians. That view is now fairly outdated. The discs are thought to be mostly holdfast structures of the frondose organisms, and at least in the 560 to 50 million year old assemblages, mostly shallow water deposits, there were possible animals present. However, in the deeper settings, what we see instead are these frondose organisms where there's limited surface locomotion, as evidenced from those trails I showed you. And this diagram on your right is actually quite misleading. What they actually look like is probably more like this, because most of the settings were deep water and therefore below the level of the photic zone. And whether animals were present in this region here, between 580 to 560, is the key question that's being addressed at the moment in our field. So finally, the few things we need to do to try and improve our knowledge of this. Basically, the Ediacaran period was defined only in 2004 as a period in geological history. The first fossils were only really discovered and documented in detail in the 1940s and 50s. And then there was a hiatus of around 20 years where not very much research went on a global scale. So it's only in the last 15 or 20 years that people have really got to grips with this period, and therefore it is one of the most dynamic research areas in paleontology. One of the first things we want to do is try and work out the relationships between each of these fossils. As it's hard for us to work out the relationships between them and modern organisms, we may as well start with what we've got in Ediacaran alone. And so that's what we're trying to do. We can then test hypotheses of how they might be related to one another. And that's going to be done by looking for new sites. This is an example in Newfoundland of one we're looking at, where all the red dots you can see are sites documented about 5 years ago in the only paper to be published on this region. We've doubled the number of sites in that area alone just by detailed looking and mapping of the sections. And also looking for new organisms. So for example, these two here, this one was actually found by Joe sitting on the front row in September, and is a totally new species to science. So there's a lot more we need to do in these settings. And the way my group has mostly been looking at it is to go at it from an evolutionary perspective, work out how they're related to each other on the basis of their shape and structure of the branching in these frondose organisms. And from that we can classify how we should be defining these as taxa, as species, and as genera, and try and work out from the bottom up what these things might have been. And it's important because, as I said, being a young period, the Ediacaran at the moment only has a top and a base. Nothing else about it has been characterized and defined. It hasn't been split up. It's 90 million years of just one period with no stages or anything else. And this paper came out last month and is the first attempt at maybe dividing up the Ediacaran period. And it's only by improving our knowledge of all these different fossil assemblages around the world, of the chemistry of these environments and how it relates to the glacial events and global climatic events, that we'll be able to do this and understand better how the first animals evolved. So, the Ediacaran period was a time of immense physical and biological change, with a lot of the organisms that are very different to anything we see alive today. But they do record the initial evolution of modern marine environments. And finally, to finish, there's lots of fascinating questions that remain to be resolved in this period. I'd encourage anyone who's interested in paleontology at all to definitely consider looking at this, because these big questions are where a lot of work remains to be done. So finally, I'd just like to thank my main co-workers, my two PhD supervisors, Duncan in Canada and Martin in Oxford, my two most recent field assistants, Joe and Jack, and thank you all for listening.
I'm sure we have lots of questions. Who wants to go first?
You said one mechanism might be the increase of oxygen.
Yes.
What do you think are the other trigger mechanisms?
The oxygen one has been the traditional one, because everyone has assumed that from the start that these were animals and therefore would need oxygen. And as soon as it was noticed in the geochemistry records that oxygen did rise at this point, everyone jumped on the bandwagon and said, yes, these must be related. My PhD was originally meant to be on trying to combine and correlate the exact first appearance of these organisms with the exact first rise in oxygen. And we found after maybe a month or two that it wasn't possible to do that. We couldn't just assume that these organisms were using oxygen, because we can't be certain that they're animals. There's nothing about their morphology that suggests that they had to be using oxygen. And so there's a variety of different ideas being bandied about. Some are intrinsic, that maybe there's a genetic mutation that just happens to happen at this point in time in Hox genes, for example, if you're familiar with those, that would allow a lot of diversity to suddenly evolve without any external cause or factor. There's also suggestions that they may have been feeding off completely different things. So I'm involved in a group that's looking at the possibility they might have been symbiotropic and feeding off some of the chemicals, sulfur or iron, that were rich in these ediacarinations, but that then disappeared when you did get the rise in oxygen later.
Anyone else?
I had thought that the fractal form of these biota meant that it couldn't develop an out or whatever. Therefore, it's a dead end. But you're suggesting actually these weren't working. Just expand that.
Yes, it's a good point. That's one possibility, and I think what I would say is that if these are true animals of fractal forms, they wouldn't have evolved, I don't think, into the groups that we see today with mouths and guts, 3 guts, anuses. They're things that would have evolved into nidarians, such as the sea pens, or into totally different groups. My personal opinion is that they are either going to be very basic animals, which are part of a group that others within that group then evolved into the main body of animals, or that they may be algae. There's a lot of deep marine algae that don't photosynthesize that people around here are actually finding that are very little studied. Or that, as one of the authors I quoted there, Silacker, suggested, that these are a completely extinct kingdom, that, as you say, fractals is a dead end. It's a very good way of getting nutrients and increasing your surface area with respect to your volume. It's something that a lot of organisms, plants for example, vascular plants, have utilised as a way of growing to get access to light or to oxygen. But it doesn't necessarily mean that they're animals. That could be just conversion.
Yep.
Going on from the idea that they might be a dead end, does the volcanic ash play a significance to possibly extinction as far as that?
I don't think that the volcanic ashes could be linked to an extinction. What we get is hundreds and hundreds of these fossil-bearing surfaces, and each one of them has been preserved by an individual volcanic event. And therefore, what we're actually seeing is that when these assemblages stop, when we don't see any more fossils, it's not necessarily because the organisms all went extinct and died out at that time. It's just because volcanism stopped in that region. And as soon as you stop getting ashfalls, you remove the way in which these fossils can be preserved, and therefore you're not going to see them again. So rather than the volcanoes being possibly the cause of an extinction, I think that the lack of volcanoes is showing us what appears to be an extinction that really isn't necessarily there. If we look at this is based on Newfoundland. If we look in Australia, in rocks maybe 5 million years younger, we see some of the same forms. So they didn't go extinct globally, it's just a regional event.
Okay, I have one last question. What fossil would you be most excited about to find, personally?
I know it sounds very dull, but if we were to find a true sponge in the Ediacaran, they're meant to be the most primitive animal fossils, the ones with the fewest morphological characters, and therefore the ones that would be the most basal. If we were to find something that